Okay, so good afternoon to everybody, or actually good morning to our speaker from New York, and to some of our guests today joining us from the US and maybe elsewhere. Welcome. I'm really delighted that we have a very special guest who is launching our 2022 lecture series today. Briana Bibel, or Bri, is a very enthusiastic and active young scientist who has recently completed her doctoral work at the Cold Spring Harbor Laboratory in the Joshua-Tor group, and she will soon be joining the Fujimori group at the University of California in San Francisco as a postdoctoral researcher. And for her PhD, Bri has been using biochemical, biophysical, and structural approaches in vitro to try to characterize how the RNA interference pathway, RNAi, regulates gene expression, and more specifically, how the actual interference complex works mechanistically at the molecular level. And she's going to be talking about this work today for us at her seminar. As we were saying before, some of you may know Bri from her highly educational and quite entertaining site, the Bumbling Biochemist, which we highly recommend, and also from her Twitter account, in which she acts as the student ambassador of the International Union of Biochemistry and Molecular Biology, the IUBMB. And I will be posting again this information for everybody at the chat. As you know, our society has recently joined the IUBMB as a member or adherent body, as it's called, from Cyprus. And so it is really a great pleasure to welcome you, Bri, IUBMB student ambassador here today. Thank you for accepting our invitation. Bri's seminar title is Regulating the Regulator, Phosphorylation-mediated regulation of the RNAi effector protein Argonaute. And so without further ado, we are looking forward to your seminar. And so the floor is now yours, Bri. Thanks.
Thank you so much for inviting me and for your kind words. And thank you to everybody who has come. Can everybody hear me okay? Okay, awesome. Okay, so I want to tell you about my work studying this protein Argonaute, or AGO, which, as I'll show you, is the effector protein of the RNA interference, or RNAi pathway. And so I've been studying how phosphorylation of Argonaute helps regulate Argonaute and influences its ability to regulate the protein production at the level of messenger RNAs. So RNA interference, or RNAi, is an evolutionarily conserved way in which organisms control the level of proteins in a sequence-specific manner. So it involves an Argonaute or an AGO protein that's bound to a small RNA, which can be something like a microRNA or an siRNA. And this small RNA is going to direct Argonaute to the site, to messenger RNA target sites that share complementarity to the small RNA, decreasing the production, the amounts of the messenger RNAs and the production of their corresponding proteins. And so this is a method that our cells use in order to control the expression of the majority of our genes. And it's also a powerful experimental tool because of the sequence-specific nature. So it's used a lot in labs for knocking down the expression of genes to reduce the protein production without actually messing with the actual gene. So in a little more detail, so AGO binds to a small RNA, which can be a microRNA or an siRNA, to form a core RNA-induced silencing complex, or RISC. These small RNAs are around 22 nucleotides long, and at their 5' ends, they have a 6 to 8 nucleotide long seed sequence. And this is going to allow AGO to use them as guides to find and bind messenger RNA targets that contain complementarity to the seed and variable amounts of downstream complementarity. So these sites are typically located in the 3' UTR of the targets. And upon binding, AGO is able to, alone or with the help of cofactors, as I'll get into, induce translational repression and/or mRNA degradation, reducing the amount of the corresponding protein that's made. What's really cool about this is that once AGO releases the target, it's still bound to the guide because the core risk complex, AGO plus guide, is incredibly stable. It has a half-life of weeks. So as long as AGO is able to efficiently release this target, then this core risk complex can be used to target additional messenger RNAs that contain complementarity. So this gives this core risk complex a lot of power directed towards the messenger RNAs that correspond to the small RNA. So where do these small RNAs come from? So microRNAs, they get transcribed in the nucleus. They have genes just like all of the proteins do, except that in the case of the microRNAs, they're going to function at the RNA level, but they need some processing first. This processing is going to happen in a couple steps. So the microRNA is transcribed as this long precursor primary microRNA. Then in the nucleus, this is going to get cut to give you a long hairpin pre-microRNA, which gets exported into the cytoplasm. Once it's out here, it's going to bind to a dicer, which is another nuclease. And this is also going to be the point at which double-stranded RNA can come in to serve as precursors for small interfering RNAs or siRNAs. So invertebrates and plants use this siRNA strategy for an immune defense. So like an antiviral strategy, they actually used double-stranded RNA from viruses to create siRNAs that targets the viral sequences. So mammals, we have evolved more complex immune systems, and so we still really use antiviral RNAi to any large extent. But we do have the capability of processing such siRNA precursors into functional siRNAs, allowing it to be used for like therapeutics and experimental techniques. But so dicer, either from the siRNA precursor or from the pre-microRNA, what it's going to do is it's going to basically measure out a 22 nucleotide long duplex that's going to get loaded into AGO to form the pre-risk. At this point, AGO is going to eject one of the strands. AGO releases the passenger strand to form the mature risk complex. And now this is ready to go seek out targets messenger RNAs that contain complementarity to the seed region and variable amounts of downstream complementarity. What happens now is going to depend in part on the Argonaute and in part on the amount of complementarity between the guide and the target. So if there's full complementarity, such as with siRNAs, and it's in an Argonaute that is capable of what we call slicing, so this includes our main human Argonaute, AGO2, what happens is AGO can slice the target, which means that it cleaves across from nucleotides 10 and 11 of the guide. And this is going to free up the ends for exonuclease chewing, getting the mRNA degraded and leading to target repression. If, however, the target is only partially complementary, and so this is the case with animal microRNAs, now AGO can't slice the target, so instead it calls in cofactors. So it binds to a scaffolding protein of the GW182 family and brings in decapping and deadenylation complexes. This leads to mRNA degradation, translational inhibition, and sequestration. And once the complex releases the target, now the so how this release occurs, however, has been a bit of a mystery, especially in those cases where you only have partial complementarity, and so you don't have the slicing to help accelerate the process. However, a few years ago, we got some information that this release process might be regulated, at least in part, through phosphorylation of a region of Argonaute called the eukaryotic insertion, or EI. And so to show you where this is, I need to introduce you to the structure of Argonaute. So this is the structure of human AGO2, so our main AGO protein. And in this case, it's bound to a guide microRNA miR20. So like all Argonaute proteins, it has 4 main domains, N, PAS, MID, and PIWI, as well as 2 linker domains, L1 and L2. The small RNA wends its way through AGO in an RNA binding channel with the 5' ends held tightly in a pocket created by the mid-PIWI domains and the 3' ends held in the PAS domain. And so in our lab, we like to think about AGO as looking kind of like a duck. And so in this analogy, the PAS domain is like the beak of the duck biting down on the 3' ends. So where's the eukaryotic insertion? Unfortunately, I can't actually show you it in the structural model because the region remains unresolved in all crystal structures of AGO proteins. However, it's located towards the very C terminus of the protein. And so in this region, which I will depict in dotted lines. We do know some things about the eukaryotic insertion, however. A few years ago, it was found that, it contains a cluster of potential phosphorylation sites, 4 serines and a threonine in AGO2. And a few years ago, it was found that these sites get phosphorylated in a variety of tissue types and organisms. The Mendel lab determined that this phosphorylation was carried out by the kinase CK1 alpha, and the phosphorylation could be removed by the phosphatase PB6. These two were engaged in a phosphorylation-dephosphorylation cycling that appeared critical for maintaining proper RNA interference. So if they had a phosphomimetic where they mimic the fully phosphorylated state, 5xe, or a phosphonull where they mutated those 5 sites to alanine so they couldn't get phosphorylated, they got global dysregulation of microRNA-mediated repression, but in different ways. So with their phosphomimetic, they found that when they pulled AGO out and looked at what it was bound to fewer messenger RNA targets. Whereas with their 5XA, it was bound to a wider range of targets, but with lower individual coverage of each, like AGO was getting stuck. And so these findings implicated this phosphorylation cycling as being important in regulating AGO's interaction with target mRNAs. And I decided to take on looking at this from a very mechanistic level. The key questions I wanted to answer were, what triggers that phosphorylation? Which residues get phosphorylated? And what effects does all that have on AGO's activity? So to start to answer these questions, I established an in vitro kinase assay. We take a recombinant human AGO2, bind it to a microRNA guide, and then to a complementary target. So for these experiments, I started with miR200 as my guide, and then as the target, I have a 30 nucleotide long sequence corresponding to a natural binding site in the transcription factor ZT1, which I'll abbreviate ZT1. I could then introduce the kinase and radio-labeled ATP to AGO when it's either in this RNA-free, guide-bound, or guide plus target-bound form, and look for phosphorylation using scintillation counting experiments. So on the left, I am showing you an SDS page gel, and on the right, I'm showing you those scintillation-based methods, which is where I did all my quantification. I just want to show you on the SDS page gel, just so you can get an idea of what it looks like. So basically, what did it look like? Well, when I took that RNA-free AGO and I added the kinase, well, nothing really happened. I didn't see any phosphorylation. And I still didn't see any phosphorylation when I added the guide. However, when I added the guide and the target, then I saw phosphorylation of AGO. And crucially, this phosphorylation was happening in the eukaryotic insertion because when I had did the same thing with the 5XA mutant, so it can't get phosphorylated in the eukaryotic insertion, I didn't detect any phosphorylation. But where in the eukaryotic insertion? Because remember, there are those 5 sites, and you can have different numbers be phosphorylated, which is a lot of possible combinations. And so I wanted to narrow it down. So I started by asking which can get phosphorylated. So I made what I call single O mutants, which have a single site open for phosphorylation. So one of them is serine and the other 4 are mutated to alanines. I could do this for each of the possible sites and then look to see where we saw phosphorylation. When we did this, we found that we could only see phosphorylation when 828 was open and available for phosphorylation. So this was telling us that only 828 could be phosphorylated in the absence of priming phosphorylation. And so what I mean by this is that CK1 alpha prefers to phosphorylate a serine or a threonine 3 residues downstream of a previously phosphorylated, like what we call priming site. And so I wanted to see whether 828 phosphorylation could prime for further phosphorylation. So now I made what I call double O mutants. And so these have 2 potential phosphorylation sites open, 828 and one other site. If we saw an increase in phosphorylation when in one of these double O combinations compared to 828 alone, then we would be able to say that 828 was likely priming for the second site. And when we did this, we saw an increase in phosphorylation with 831 open, suggesting that 828 primes for 831 phosphorylation, consistent with CK1 alpha's known preferences. We then tested if that could prime for the phosphorylation of 834, and we found increased phosphorylation again with our triple O mutants. Although we also think that 824 might play a role. And it's more heterogeneous after that initial 2 phosphorylations. So we know what triggers it and where it's getting phosphorylated, but what effects does this actually have on AGO's activity? And more specifically, what effects does it have on AGO's ability to interact with target RNAs? So to investigate this, I established a target release assay. So we take AGO, load it with the guide and with a target, but in this case, the target is radio labeled. Maria, please stop drawing on the screen. So in this case, we bind AGO to the radio labeled target, and then we add an excess of unlabeled target. And then we track the release of the radio labeled target over time using a slop blot filter binding assay. So in these experiments, I have a nitrocellulose membrane on top and an RNA, a nylon RNA binding membrane on the bottom. This way, when I flow the time point mixture through, then I will be able to see the protein and any bound RNA is going to get trapped on the nitrocellulose membrane. The free RNA will flow through, and I can then compare the amounts. So when I did this, and then I could add the kinase and see if I see a difference. And so when I did this, I found that phosphorylation greatly increased the off rate, suggesting that phosphorylation was promoting the target release. And this was happening in a eukaryotic insertion specific manner because our 5XA mutant that can't get phosphorylated in those sites, we don't see any effect of the phosphorylation. We also found that the effects of phosphorylation are additive. So the more sites we have open, the bigger the effect on the off rate. And this suggested to us that it might be an electrostatic mechanism, some charge repulsion. So although we can't see the eukaryotic insertion in the crystal structures, we see that it seems to be pointing out towards the solvent exposed backbone of the target. You're introducing negatively charged phosphate groups across from the negative charge of the target backbone, which we hypothesized would lead them to repel each other, promoting target release. To test this, we repeated the experiment under high salt conditions. And we found that the effect of phosphorylation on rate basically disappeared, suggesting that this is indeed an electrostatic mechanism at play. This leads to a mechanistic model whereby AGO binds to a guide and then binds to a complementary target. This triggers the phosphorylation by CK1 alpha of 828, followed by a hierarchical phosphorylation. then have the negative charges clashing, leading the target to be released. This would then, AGO would then have further, would be inhibited from further target binding. However, the system could quickly be reset by the phosphatase PB6 in cells. This would reset the system. But then, of course, when AGO binds a new target, then CK1 alpha can come back. And so this leads to the still kind of outstanding question of how the target is actually promoting the phosphorylation. So we wanted to figure out the minimal target requirements for triggering this phosphorylation. So we took that 30 nucleotide long target that we have been using and started shortening it from the 5' ends. We saw a dramatic decrease in phosphorylation when we went below 14 nucleotides. And this suggested to us that supplemental region pairing might be important. So the seed region is the main region, the main way that full complementarity to the seed region is the main way that targets are basically targeted, but you can also have additional pairing, and this supplemental pairing is often centered around this 3' supplemental region of around 12 to 17 of the guide. And so we wanted to see whether supplemental region pairing really was important. And so we repeated those experiments, but we had a, with the full length target, except in this case, we tested a target that only matched in the seed region, and we saw very little phosphorylation, suggesting that this complementarity in the supplemental region was important. However, a bit to our surprise, we found that it wasn't just having pairing in that region that was important, it was actually that seed plus supplemental layout. So if we had a target that was fully complementary, like a siRNA-like one, we barely saw any phosphorylation there, even when we used a catalytically inactivated AGO2 so that we prevent any confounding factors from AGO actually slicing the target. These and other findings suggested to us that there might be a conformational change that was important for promoting the phosphorylation. So we know from crystal structures of AGO bound to various targets that it has to undergo a conformational change in order to accommodate pairing in that supplemental region. This is involves movement of the PAS domain away from the region of the eukaryotic insertion, which could make the region more accessible. So we know that these changes happen upon target binding, and with the seed and supplemental pairing in particular. However, we don't know if anything is changing in the eukaryotic insertion itself that might help explain how the target binding was triggering this phosphorylation. So in order to get structural information, we turn to hydrogen deuterium exchange mass spectrometry, or HDXMS, which was carried out by our collaborators at UC San Diego, Elizabeth Combes and Steve Saletti. And so for anyone who's unfamiliar with this technique, it's a way that you can assess secondary structure and solvent accessibility of regions of a protein in solution. So the basics of how it works is you start with a protein in a water-based buffer, and you label it by diluting it in deuterated buffer. Hydrogens in the protein backbone can exchange for deuterium and labeling the protein. You can then quench at different time points and then do proteolytic digestion and mass spectrometry to measure the weight of those pieces. And if they have exchanged for deuterium, they'll be heavier. And importantly, the observable exchange is only going to come from the backbone amide hydrogens. So the hydrogens attached to the carbons are not going to exchange on these time scales, and others are too labile that they will exchange back when you do the quench step. And so this is going to tell you about the backbone structure, because these regions can only uptake deuterium if they are at least temporarily free from hydrogen bonding interactions that would have them tied up. And so therefore regions with a strong secondary structure are going to uptake very slowly, if at all, whereas regions that are flexible and solvent exposed, they're going to take up deuterium rapidly and to a large extent. That is, of course, unless there's something bound there that is occupying those hydrogens, and then you can see protection from exchange. So you can compare proteins in different states and get information about where things might be bound and where you have some conformational changes occurring. So we wanted to see whether we could detect differences in the eukaryotic insertion for AGO in the RNA-free, guide-bound, or guide plus target-bound states. And so we all of those states, and I want to show you what we found. You're going to see blue, and you can see that you see a lot of blue. And so you can see that we have clear evidence of the 5' binding site where it has that tight interaction. You see great protection in the guide and guide plus target bound states, as well as you can see the evidence of the 3' binding. In addition to those specific sites, you can see that you get a great protection over most of the protein. And this is really consistent with past results of limited proteolysis experiments. So we don't have any structures of RNA-free AGO. However, in our lab, and I repeat these experiments, we found that the RNA-free AGO is incredibly sensitive to proteolytic digestion, whereas once you bind it to a guide, it's really incredibly stable. So like 60 minutes with thermolysin, and it's still hard as a rock. And so this is really consistent with what we're seeing with the HDX results. And so here, this is showing you the different peptides, showing their uptake. And so the ones that are in blue have significant protection upon the guide binding. And you can see that these peptides are really almost all of the peptides, and they're really localized all throughout the protein. And we know from structures that the guide RNA actually contacts all of the different domains. So this is very consistent. But what about target binding? So with target binding, we find that the effects are much more subtle. So here I'm actually showing you, I bumped up the scaling. So here the changes, the deepest blue is corresponding to a 25% protection offered by target the guide alone, and the red would be the opposite way. As opposed to what those dramatic effects that we saw with the guide binding, here we find that the changes are more localized, but we do see the evidence of the changes we would expect based on the crystal structures, such as in the L1 and helix 7 regions, which are known to move as part of that movement to accommodate the supplemental region pairing. So all of this was really exciting to see, and it helped confirm some of our suspicions based on the crystal structures and things. But what about the eukaryotic insertion itself? Because that's what we're really interested in. And so we didn't really see much of anything. And so here, because this region is unresolved in the crystal structures, I've modeled it in. And you can see that in the shortest peptide covering this region, 812 to 842, we don't see any, we barely see any difference between any of these states, and we see no difference between the guide and the guide plus target. We do see a little increased uptake in the RNA-free. However, if we take the overlapping peptides into account, we can see that this increase is actually coming from this portion of that peptide that was C-terminal to the phosphorylation sites, suggesting that nothing in the actual region containing the phosphorylation sites had any changes that were observable with this technique. So it's important to note that there are lots of types of changes that wouldn't be detected with this method, but we don't see any evidence of there being any introduction or change in the strong secondary structure or binding interactions with the RNA or that sort of thing. So we don't see any evidence of changes in the eukaryotic insertion being responsible for triggering this phosphorylation. We also don't see any changes in like the surrounding region that could help explain this. And so if we go back to thinking about how target binding is triggering the phosphorylation, we don't see any changes in or around the EI that could help explain this from the HDX data. So we talked about from the crystallography data how we know that the PAS domain is moving away from this region and might be making it more accessible. However, we saw that we had really great accessibility of that region. We had high uptake in basically all of them. The RNA-free, the guide-bound, and the guide-plus target, they all showed a high degree of uptake, suggesting that this is like a flexible, loopy, solvent-exposed region. And with the RNA-free in particular, we saw that there was high uptake all throughout the protein. And so if it was just an accessibility issue, we would think that the eukaryotic would be very accessible for phosphorylation, and yet we don't see phosphorylation of the RNA-free AGO. And so it must be something else. And so what if it's actually the importance of the target binding isn't that it's making this region change structurally, but that it's actually the target itself. And so the reason I say this, so CK1 alpha is an acidophilic kinase. It has these basic patches or anion binding sites on its surface. And we hypothesized that the target RNA, the negative charge backbone of the target RNA, is actually interacting directly with the but we know that CK1 alpha greatly prefers to phosphorylate a prime site. It's known that in some cases, a cluster of acidic residues downstream of the phosphorylation site can kind of play the role of a priming site and compensate and bind to those anion binding sites and position the phosphorylation, the site to be phosphorylated in the active site pocket, which is shown to be the importance of having that priming. So there have been some suggestions that these that acidic residues upstream of this sequence in the eukaryotic insertion might serve a priming role. However, when we mutated those residues to neutralize them, and we didn't see any effect on the phosphorylation, suggesting that this cluster wasn't playing a priming role, but leaving open the possibility that a phosphate on the target backbone might actually be serving as a priming phosphate. So now if we go back to that mechanistic model, we think that target binding is leading to a conformational change that's going to position AGO or the RNA to interact with and potentially prime CK1 alpha. And so then you have the phosphorylation, you have the target release, and you're inhibited from further target binding. And then the system gets reset and you do it all again. And so this might seem like a really inefficient method of regulating things. But we think it's actually a very kind of clever strategy that cells can use to optimize the balance between target site affinity and the risk of having your risk stuck onto targets. And so you could think that if AGO had partial complement, if it had supplemental region pairing, this would give it higher affinity as well as the extra specificity you get from being able to differentiate between different target sites. But then you could potentially have to worry about AGO getting stuck on those sites because you don't have the splicing to help accelerate the release. So if this was the case, we would also expect that this would be less, this mechanism would be less like important for siRNAs where you have like slicing. And in fact, if we go back to those results we saw before, we saw that the siRNA like targets didn't even trigger the phosphorylation. And since I couldn't get it to be really phosphorylated, I tested phosphomimics, and those didn't seem to affect the ability of AGO to slice the targets. Further evidence for this being a microRNA specific mechanism is found in the evolutionary conservation. So this eukaryotic insertion is highly conserved among eukaryotic AGO proteins that specialize in dealing with microRNAs, but not other AGO proteins. And so this is clearest to see in Drosophila, where they have two AGO proteins, AGO1 and AGO2. And AGO1 handles their microRNAs, whereas AGO2 specializes in siRNAs. AGO1 has those phosphorylation sites in the eukaryotic insertion, and AGO2 doesn't have that eukaryotic insertion. You also don't see this in other types of Argonaute proteins. if we look at the human Argonauts, we see that all of them have these sites. So there's only the threonine is only conserved in AGO2, but the 4 serines are conserved throughout all these AGO proteins, including the human AGO proteins. And this suggests that it might be a more widespread mechanism. And in fact, we also found that the other human AGO proteins were susceptible to target-triggered phosphorylation. And so overall, we have this mechanism that we think plays a role in helping with the recycling of this risk complex, freeing AGO that is potentially had the potential of getting stuck on a target due to having that higher affinity and but also that specificity. So you have increased efficacy of those sites, but you don't want to lose efficiency by having AGO stuck on those sites. And so this is where phosphorylation can come into play. And so if anybody is interested in learning more, we have our results were recently published, or not, sorry, our results were recently posted on bioRxiv as a preprint that you can check out, and I'd be happy to answer any questions. But first, I'd like to thank all the other people who made this possible. So thanks to everybody in the Joshua-Tor lab, especially my PI, Leemor Joshua-Tor, Alado Kayam, who was a research scientist in the lab who really helped mentor me and was an amazing mentor. John Ipsaro, who helped me with some of the HDX analyzing, as well as our amazing lab techs, John and Soraya, who have since moved on. And our new lab techs are really awesome too. Simati and Matthew, but John and Soraya really did a lot of expression and cloning, and so I'm really grateful. Also like thank my thesis committee and funding. And now I'll be happy to answer any of your questions, and I apologize for those disruptions.
Thank you so much, Bri, and we apologize for this. I couldn't have imagined that giving the Zoom address would cause this. I'm really sorry for the stress. Anyway, your talk was really fascinating. I mean, it's great how you could use all the different techniques to get into such a detailed mechanistic insight. It's extremely interesting. Congratulations.
Thank you.
Are there questions from the audience? Please just unmute your microphone and put your question.
So can I ask something very, I might miss it. First of all, Brianna, thank you so much for this brilliant talk and for the brilliant presentation. So as Niovi said, deep into the mechanisms, but your slides were very helpful to understand all this complicated structural work. So just I might miss it. So am I correct that in humans we have 4 AGO proteins, correct? So what's the difference between each one of them? How does their function differentiate, AGO1 to AGO4?
So there is not, they are incredibly similar structure-wise and sequence-wise. There is not any clear distinctions between most of them in terms of they all seem to bind the same sort of things. There have been different functions, differences in some of them that, like papers will say, oh, this one's important for this, but there's not really any clear differences. The clearest difference is that AGO2 is the only one that has that clear slicer ability, the ability to slice targets. So there's been some recent work that AGO3 can actually slice target like certain targets, but it has to be bound to like a really short guide RNA. There's some, it's not quite clear, but the other AGO proteins, so AGO 1 and 4, they actually have like amino acid substitutions that would prevent in their catalytic residues, so they wouldn't expect them to slice. AGO2 is also like the most abundantly expressed. AGO4 is the reason why I didn't test it is because it is very, very low expression, both in cells and if you try to express it recombinantly. But there doesn't seem to be any sort of clear distinction, and they can compensate for the loss of others. There's a couple rare instances. So AGO2 is actually required for the processing of a non-canonical microRNA. And so MIR451, it's actually, it bypasses the dicer step and gets cleaved directly by AGO2. And so you need AGO2 in order to do that processing step. And that's really important for like blood cell development. Yeah, so sorry, that was probably way more than you wanted to know. But...
No, brilliant. Thank you. Thank you so much. Yes.
Can I ask a follow-up question on this? You mentioned the protein phosphatase PP6. Do we know of any other phosphatases that do the dephosphorylation step so that Argonaute can get reactivated for a new round of processing? And I mean, how do you orchestrate these different activities of getting the phosphorylation to start with and then at the end removing it with the phosphatases? Do we know anything about how these are coordinated?
Yeah, so there's only been that one paper, which was kind of like the start of my whole project. So from the Mendel lab, and they kind of discovered this phosphorylation cycling. They were using a CRISPR strain to try to identify regulators of the RNA interference pathway. And they found that the phosphatase, that this PP6 phosphatase was important for in their functional screen of like a reporter-based assay for measuring the RNAi. And so they found that their knockdown of this phosphatase was causing problems with this RNAi. And so then when they looked deeper into it, that's how they discovered this whole phosphorylation cycling. They didn't find any other phosphatases that could help, that could explain things. And they seemed to get pretty full response from knocking out the phosphatase. But of course, there are possibilities that there are other phosphatases or kinases that could play a supporting role. However, when they knocked out like either of them, they saw basically the things that you would expect if it was just those. But as terms of like all of my work is done totally in vitro. I haven't done like any cellular work. And so the temporal dynamics and spatial dynamics within cells and all of that is still something that needs to be worked out. That's just not the kind of experiments that our lab is really set up to do.
Mm-hmm. Yeah.
Naomi, can I ask a question to Bree, if that's okay?
Of course.
Bree, first of all, thank you very much for this wonderful talk. You really used sophisticated in vitro techniques to delineate the molecular mechanisms of your protein and interactions and phosphorylation. I congratulate you on that, first of all. And I would like to ask, so on the structure of your protein, you showed how that I guess you said duck's head was sort of moving. Was anybody able to demonstrate that sort of the movement within the protein itself using techniques like maybe FRET or similar experimental techniques to delineate those movements that might sort of give you really helpful ideas about those exact functioning of the protein? And the second,
Oops. Oh, did we lose something?
They're left?
I don't know. It seems maybe she lost her connection.
Yes, maybe.
Maybe she will try to rejoin. Maybe Bree, let's hold on to the answer in case she can join us again. Are there any more questions in the meantime while you are waiting for Ender to, here she comes, okay. Hello, Ender. We lost
hi.
you. Yes, okay.
I do not know what happened. I
Okay,
apologize.
glad you're back. So
Yes.
you wanted to continue your question?
Yeah, so I had that question and another one that I was curious about. You said, Bree, that part of your protein was disordered in the crystal structure. A lot of these reactions happen in solution inside the cells. So I was wondering if anybody tried to get the structure using maybe NMR, like a solution structure of your protein, which might actually also give you a little bit more realistic ideas about like the mechanisms of functioning of your protein as well. Those were my two questions for you. And again, thank you for this wonderful talk.
Well, thank you so much. And so basically there were 1 or 2 papers that did some FRET early on. I would have to go back to them, but I haven't seen anything using FRET recently. But that's a good reminder to go and look back. Thank you. As for the NMR, this protein is like 97 kilodaltons. So I don't know how realistic that would be in terms of getting an NMR structure. But yes, that's kind of why we turned to the HDX, was to get solution phase information.
Yeah, that makes sense, because that large of a protein NMR would not be applicable. Yeah. Thank you so much.
Thank you.
Any more questions from anybody?
Hi, Brianna. I have a quick question. If that's okay. So first of all, thanks. A very nice talk. Fascinating to see what you have done, and congratulations actually getting your PhD and the new position.
Thank you.
So probably I missed that, but when you mentioned the phosphorylation sites of the 4 serines and 1 threonine, so those they are basically phosphorylated by a number of kinases or by a specific kinase. Can you just clarify that, please?
Yeah, so all of this I was doing with a single kinase, CK1 alpha, but it does hierarchical phosphorylation. So once it phosphorylates one site, then it can use that to prime for further phosphorylation of the additional sites. But all of the phosphorylation was actually taking place with this single kinase.
Okay, thanks. So there's no evidence of any other kinases competing that specific kinase?
No, there was. I mean, there is still open the possibility that there could be another kinase playing some sort of role in potentially phosphorylating like some of these sites, and then CK1 alpha could act, or CK1 alpha acts and then these other sites. But this was the only kinase that was identified in the original screen, and we found that CK1 alpha alone is at least capable of doing this in vitro without any other kinases being involved.
Okay, thank you. Thanks a lot.
So Bree, will you be continuing this line of research as a postdoc at UCSF, or you'll start another project?
I'm going to be doing a totally different project, some things with ribosomes, so really excited.
Okay. Are there any other questions? Let me make a very quick announcement. We would like to bring your attention to a very interesting 2-day conference that will be taking place at the University of Leeds in April. I just put in the chat the link. The Nobel laureate Richard Henderson, who got his prize, he shared it for cryo-electron microscopy, will give the plenary session. And the event is open for free for anybody, but you need to register in advance, and the link is given in the chat. So it would be a good thing to have in mind. So, Bree, thank you so much. I really, again, apologize for the disruptions. We enjoyed your talk, and also we enjoyed meeting you. I wish you all the best in finishing and publishing this work and for your postdoc. And it was a real honor and a pleasure to have you here. Thank you.
Thank you so much. It was really great. Thank you all, and thank you for the great questions.
Thank you.
Great meeting you. Keep up the good work with science communication. Please
Thank
do.
you so much.
Thank you very much, Bri. Thank
My
you.
pleasure. My pleasure.
And thank you for all the participants. Okay, good night, everybody. Bye-bye.